Hi again, higher chemistry students, and today we're going to continue with Unit 2, Nature's Chemistry, Section F, Proteins. Proteins are the major structural materials of animal tissue and are also involved in the maintenance and regulation of life processes. Plants and animals need a supply of nitrogen to build these proteins, and for us humans, they are an essential part of a healthy diet as they are required for growth of both muscle tissue and they aid repair. So proteins are molecules that make up the muscle fibres, your hair, your skin, your nails, the collagen, the hemoglobin in your blood, the enzymes that keep your body functioning healthy, and they are very large molecules or polymers made by joining many small molecules or monomers called amino acids together. Amino acids join together to make these large natural polymers or your proteins via a condensation reaction. There are many different proteins, and proteins can be specific to whatever a body needs at a particular point in time. Our bodies cannot make the proteins, therefore we must eat the protein in our diet to gain the essential amino acids that our bodies need to function properly. So amino acids are small molecules. They contain two functional groups, an amine or amino group and a carboxylic acid group. Both of these groups appear on the same molecule. In between both of these functional groups can be pretty much anything within reason. So where we see the X on the diagram on the slide, we can replace the X with hydrogen, with an alkyl group, by that I mean a CH3, C2H5, or any other alkyl group. We can even replace it with some sulfur, phosphorus, or other nitrogen atoms. It is possible to get many different isomers of proteins and amino acids, but when you have your two specific different groups, your amino group and your acid group, bonded to the same carbon atom, we call these specifically alpha amino acids. Here are two alpha amino acids. There are about 20 naturally occurring amino acids, and the two simplest of these are glycine and aniline. When we join different amino acids together, we can form different proteins. So to form your protein, we join our different amino acids together. Here we can see three different amino acids on the slide. The green box can be anything in between we've spoken about in previous slides. We join the amino acids together by removing an H from the amine group and OH from the carboxyl group. In doing this, we remove water, and therefore we can join together what's left behind from our amino acids. When they link together, we form part of your protein chain, and we form a new linkage known as an amide or peptide link, which is a C double bond O NH group. When we eat our proteins, we obviously digest our food, or we hydrolyse the protein. When the protein breaks down, it breaks back into the small amino acid molecules. These can easily pass into the bloodstream. And this hydrolysis, this breakdown, is carried out using either an acid that acts as a catalyst or an enzyme, which is a biological catalyst. So when we break down our protein, we always break it in the same place. We identify the amide or peptide link, your C double bond O NH, and we always break the bond between the carbon and the nitrogen. In doing this, we form our separate small amino acids. There are two different types of protein that exist. Fibrous proteins, which are long, thin structures. They basically make up things like your muscles, your tissue, your hair, the collagen. Or we have globular proteins. These are like big balls of wool or spiral chains that are compacted together, and these help to regulate the hormones and maintain a healthy body. Globular proteins include things like insulin to regulate your blood sugar level and your hemoglobin within the blood. Enzymes, as we said, are an example of proteins. Enzymes are biological catalysts, and from National 5, you know that enzymes are very specific. One enzyme for one type of reaction, they are not interchangeable, and we call this the lock and key mechanism. So for example, starch can be broken down into maltose using the enzyme amylase. Amylase helps to speed up the reaction, and we know it will not work with any other reaction due to the lock and key effect. That means the substrates will only fit into the enzyme structure, as the diagram shows. Each enzyme is specific to the reaction that it speeds up or catalyzes. Your front door key would not open your car and vice versa, the lock and key mechanism. But there are factors that can affect how well an enzyme works. The two specific ones are temperature and pH. Because enzymes are biological catalysts, they work best at what we call an optimum temperature, and this is roughly body temperature of 37 degrees. Too low below this temperature, the reaction is very slow. Too high above this temperature, you denature, therefore change the shape of the enzyme. The lock and key mechanism can't work. pH is also another factor. Some enzymes work best in acidic conditions. Others work best in alkaline conditions. So for example, catalase, which is an enzyme present in both potatoes, celery, and the liver within your body, which helps to break down the toxic hydrogen peroxide, is an example of this. We can carry out an experiment to show that optimum pH and optimum temperature give optimum results with an enzyme. We can do this by using potato, as we know it contains catalase, and test the effect of the enzyme under different pHs and different temperatures. The pH that gives the most bubbles of oxygen in a given time is known as the optimum pH, and the temperature which gives the most oxygen bubbles produced in a given time is known as the optimum temperature. Like we said, for enzymes, because they are biological catalysts, they work best at body temperature, which is approximately 37 degrees. So from this graph, you can see the optimum temperature where you're generating the most oxygen bubbles in a given time is at the peak, which is at 37 degrees Celsius. If we go too far beyond this temperature, we see a rapid decrease in the number of bubbles because we have denatured the enzyme, therefore changed its shape permanently, and it doesn't work effectively. Alternatively, we can see the optimum pH for the potato disc containing the catalase enzyme is a pH of about 7.4. Therefore, it's a slightly alkaline pH where we get its optimum effect. Can you now complete exercise 2F and enjoy?